Right, so welcome back everyone. This is day 3, brain network modeling. We have a slightly smaller audience today than we did the first time, which is certainly a version of a good thing. So I hope that the first 2 sessions, you guys got a lot out of it. Those were really the main kind of content pieces for this week. And today, what I want to do is really 2 to 3 things. Firstly, I want to field questions and feedback from people, from the homework, from the notebook that you're working through, from the lectures, from whatever random stuff that comes to mind that you think might be something worth discussing in this context. We can do it. I believe I have the power to unmute people, so we can do a bit of actual conversation, or we can field some questions through the chat. I really want to encourage you guys to, chip in, and we'll have a little bit of back and forth today. So that's item 1. Item 2 is I'm going to do a little bit of comments and just kind of feedback a little bit on the homeworks, and this is just going to be highlighting some of the submissions that I thought did a good job, and maybe actually commenting on 1 or 2 of the points that are discussed in the summaries that you guys wrote, and the approaches that you took to it. And again, that's something that we can turn into a conversational piece as far as you guys are inclined. And thirdly, and this is, I said like maybe 3. So the third thing that I thought I would maybe do a bit of is look at the other notebook that I mentioned earlier that's in the repository, and just kind of go through it briefly at a relatively high level to give an example of a different type of application of these type of tools. Okay. And I'm not going to do that for a very long time. Like, it will be a kind of light touch run through, but that will hopefully be interesting to people as well. And not necessarily aiming to go longer than we need to today either. So we'll go through these things, and then if we finish early, then all the better. People can maybe go and enjoy the sunshine. Excellent. So to the first point, let's questions come through, please. If you guys have some questions from any of the above, then just fire them in the chat as and when you're ready. We'll have a reminder from Marcello. The attendance code today for you guys to register your attendance on the website is 5. So remember that when you're going and logging the fact that you're here. Okay, good. So I'll keep my eyes on the questions if and when they come through. And in the meantime, I will just give some comments and feedback on some of the homework submissions. So I will share my screen. So apologies for last time with my incessant sneezing fit. That was almost certainly a kind of hay fever pollen thing, as I was out running just about half an hour before I gave the thing. So I guess that caught up with me. Touch wood, no sneezing fits today, but maybe I'm jinxing myself. Okay, just trying to move some things out of the way so we have a clear screen to share with you. Okay, so as you hopefully can see, because I'm screen sharing, please just fire some things in the chat if you don't see my screen. I have some Jupyter notebooks open. So we didn't get a whole load of submissions back, and I guess the guys of you who are on this call are the folks who actually put in the submissions. So hats off to all of those, and a few familiar names here. Hi, Anil, Brainhack, Denizen, and Emils. I think we had a chat at the same course last year. So nice to see some familiar names. So I'm kind of going to step through one yeah, maybe I'll just step through each of your guys' assignments and note what you guys got up to. So the first one I have here was from, it's Mr. Gao, what is it? I remember, I forget the first name, apologies, but I say Mr. Gao, was taking a look at the parcellations and did a good job. So I said, okay, do some Googling to find the what these abbreviations correspond to. And lo and behold, he did a good job of finding the right link for that. So made a note of each of these and then gave us some plots of where we can see these brain regions. No simulations actually in this submission, but you know, good job of finding the brain region abbreviations list. This next one is from, bear with me. Yep, Seshu Yanga. So good job. So Seshu, and actually a number of you were interested in doing multiple region stimulation. There's about 3 or 4 that occurred to a few of you. And Seshu made me work for this one in that you kind of gave in the submission, but it wasn't executed. Maybe it was because it was a very large file or something, but I ran your code. So I kind of reconstructed the simulations that you did. And so you were doing things like, if I just scroll up here, looking at stimulating multiple areas simultaneously, both regions that are nearby each other and regions that are far away from each other. And there are 1 or 2 that were interesting here. For example, ones that are, say, further away, like back and front. So this one here, I think we've got, yeah, frontal. This is a left prefrontal and then V2, visual cortex. And as he's kind of commenting in the description, we have these 2 waves that are kind of propagating, one from the front backwards and one from the back forwards, and then they're kind of interfering or so it's hard to say, strictly speaking, what they're doing when they intersect in the middle, but there's definitely both backward and forward wave propagation there. So that's kind of an interesting thing to look at. Moving on, Paul Jeffrey. Paul was, wait for this to load. Yeah, so Paul actually did quite a nice comprehensive set of investigations with some good write-ups. Again, looking at stimulating multiple areas. So yeah, this was something that was a common idea. Again, you know, feel free to just put some comments in the chat, guys. Like if you want to add to my summaries, I'm giving some fairly lightning summaries, and I can see that some of you spent a fair bit of time on this. But some you know, similar to what we saw in the last one, actually choosing some regions that are far away and then stimulating them simultaneously. We had, let's just pick one. Yeah, here we go. So getting 2 regions, like one at the back and one at the front being stimulated, and then also varying the stimulation amplitude, which was something that some of the other guys, and duration, which some of you guys also did. And one of them, I think it was Jeffrey Glaister, was looking into extending the size of the envelope, which I had a comment or 2 on. So skipping down. Primary motor areas. Yeah, also played around with the stimulation equation by expanding the pulse width. That was a nice idea there. So yeah, Jeffrey was focusing on left and right motor cortex stimulation, keeping it focused. Interestingly, this is, so in terms of TMS research, which is what we're really kind of aiming at here. I mean, this could be this could be electrical stimulation. It could be, you know, in principle, it could be optogenetic stimulation if you could do that in humans. But really, like this is more or less modeling TMS. Most of the things we understand about TMS, transcranial magnetic stimulation, we know about in virtue of work that was done on the motor cortex. And the main reason for that is that the motor cortex is somewhat unique in the brain, excuse me, in that you can get a direct behavioral output from the from stimulation there. And the behavioral output is because the motor cortex has motor neurons that actually project to your limbs, and particularly your arm. So one of the kind of key things that's used in TMS research is to kind of stimulate the part of the motor cortex, you know, a sub-region of the motor cortex. Here we're being quite coarse-grained, right? We're kind of stimulating all of M1, and actually that's an extreme. I mean, you couldn't actually do that experimentally, or at least it would be difficult. But you can find a particular part of it that corresponds to that includes the motor neurons that project to the hand, and then that's one of the workhorses of TMS is like seeing what changes in things like basically the little flick that you get in the hand that when you're activating the motor neurons that control the fingers, seeing how that is influenced by things like pulse duration, pulse frequency, stuff like that. So just a little kind of contextual point there. Like a lot of TMS focuses on M1, so it's a sensible thing to look at when you're looking at simulating stimulation. And yeah, so the other thing Jeffrey was doing was extending the pulse duration. So this was pretty cool, and we see in the snapshots there, but also kind of better in the video. So we've got you know, these bilateral injections into M1, and then really this, you know, big red thing in the middle here, kind of turtle shaped little man or something. That's more or less persisting for the whole pulse duration, because I think you had that for about 300 milliseconds. And then we're seeing pretty global excitation bilaterally, which isn't that surprising, given the spatial location of the motor cortex in the middle of the brain and the amount of stimulation that you're putting into it. But, you know, interesting idea to vary the pulse width. And I mean, it was pretty wild, the effects that you were getting here. But if you were to induce this much excitation experimentally, you would almost certainly cause a seizure, and you absolutely aren't allowed to do that. So, so you know, that's a good case for in silico investigations, amongst other things. Yeah, cool idea. Emils was looking at the hippocampus and the amygdala. So this was another common theme from one or two of you, was people are interested in amygdala. And yeah, some nice kind of fairly careful investigation into what these evolution of these stimulation patterns between the hippocampus and the amygdala here. And same thing from Anil, which, yeah, actually, Christopher, well, you've done a lot of work here, but you hadn't included any of the code of the figures. So, you know, good job, but would have been nice to see the code in the figures. That's what Jupyter Notebooks are for. But Anil, yeah, was also thinking about the amygdala, and there's just another little point, interesting point that I was going to add comment on here, was, okay, so again, there's a stimulation location. I mean, something to bear in mind, I talked about this in the opening lecture, because there were some comments. I think it was a question from Marcello actually about how many regions, right? What parcellation size? So you see we've got, you know, amygdala here. That's a parcel, I guess you kind of roughly guess that's maybe about 3 squared centimeters. The motor cortex, the primary motor cortex in the parcellation that we were using, that's, I mean, it's quite well anatomically defined. You can even see it on these surface images fairly well. It's like there, okay? But it was extending like a really big, long, thin thing. The point is, you know, at the parcellation scale that we're looking at here, which is roughly 100 brain regions, or you know, roughly 80 cortical regions, it's still pretty coarse grained. I mean, you know, 80 brain regions is a lot, certainly a lot to try and understand at any level of, you know, memorized understanding, but you still end up with big parcels. And that has some knock-on effects in terms of what you're seeing with these kind of stimulation effects, because you've got like big parcel sat next to big parcel sat next to big parcel that are all, sometimes they're kind of elongated shapes and so on. So they don't always have a very, you know, if they were smaller and more, you know, isotropically shaped, then the propagation patterns would probably look a little bit more natural and actually a little bit more realistic. But it's you know, you feel like it's kind of a bit of an artifact of the way that we work and the way that neuroimaging has kind of ended up at this point in time, as a way of compromising between treating every single point in the brain independently and trying to group brain regions. Is we end up with, you know, brain regions that are somewhat larger than maybe you would like and somewhat unusually shaped. In any case, I mean, amygdala is there medially, and then here Anil's sticking with the stimulation parameters, but just taking a close look and commenting on the what we get. And I thought a nice thing, well, two interesting things about what we're seeing here. One is that we're getting propagation both backwards and forwards, which, as you know, you would expect given that this is sat in the middle, but it does do you can kind of more or less see the propagation in those two directions, separating out here. You know, this is an example of the kind of thing I was saying about these, bearing in mind the shape of the regions that you're using. So this one here, I believe this is the anterior cingulate cortex. Hello, Charlotte. Welcome. Welcome, Charlotte. Anterior cingulate cortex. This whole sliver here, this is one brain region, right? So it's kind of thin, kind of long, kind of pokey. That's an example of what I was just describing. And another example would be actually the temporal lobe. So the temporal lobe parcellation, we're looking at it on the medial surface here. You can just about see when it's getting activated. We've got a long, thin slither there, and then another one underneath it that comes through. So this guy here, this long, thin one, this is the medial surface of the superior temporal gyrus. And the superior temporal gyrus, I mean, so the temporal lobe, you can subdivide the temporal lobe. There are various subdivisions of the temporal lobe. One of the main ones is superior, middle, inferior, and then you have a bunch of things on the medial side. But the superior, middle, inferior, well, the fact is the temporal lobe is basically, it's long and thin. It's kind of this, you know, little foot, if you like, on the brain there on the bottom side, on the cortex. And it really is like the part of the brain that the cortex sits on. So the temporal lobe is kind of long and thin, and then the principal subdivisions of the temporal lobe, the superior, middle, and inferior, those are kind of stacked sausages, if you like, little. We basically draw a line along the principal pole, and we don't just make it up. There's a gyrus there. There's a sulcus there. So it's kind of a natural border, but we have a long strip there that's the superior temporal gyrus, a long strip in the middle that's the middle temporal gyrus, and a long strip underneath that's the inferior temporal gyrus. So that comes out in these kind of simulations, you see. So we have this like long thin strip here. So we get our propagation backwards and forwards in this simulation here, but it does move through some of these anisotropically shaped structures for the parcellation that we're using here. So cool. That was nice. I thought the directions of the propagation, and the other part of this that was commented on by Anil is the propagation to the contralateral hemisphere, which we're seeing just for the amygdala. Okay, and that's kind of interesting. That's to some extent a bit of a limitation of the connectivity matrix that we're using here, not the parcellation, but the edges in the network. And I mentioned this earlier on. In fact, I'll bring up I have it in the, quickly go over and bring up the connectivity matrix I commented on this in the lecture, but the connections between the hemispheres in the network that we're using are a bit sparse. There's reasons for that. I mean, basically the reason for that is that a lot of the work that's gone into identifying these anatomical connections for the source of the data that's used in the default connectivity matrix in TVB, this is not actually from humans. This is from macaques. And what it is chemical track tracing. So it's like you inject some chemical dye into a part of a macaque brain, and then it moves along the fibers that are projecting from the place where you injected it, and then you kind of you look where it goes. And the way you look is you actually chop it up, literally cut it up into sections, and then see where you're finding dye after like 12 hours or something, so post-mortem. Okay. So that's very very involved work, and hats off to the people who've been doing it for a long time. It's also very expensive work, and particularly it's much more cost efficient. It's much more efficient for people who are doing that kind of work to get two hemispheres out of a brain rather than one. Okay, so in the sense that it's if you're looking at projections with intrahemispheric sites, then you can do that in both hemispheres. You can kind of do an experiment in one hemisphere and you do an experiment in the other hemisphere. If you're interested in projections to contralateral sites, then you can only do that once because you need both hemispheres. So it's a bit of a bias in the literature from the track tracing data of studies that have used this pretty involved experimental method to study contralateral projections. So as a result, these are a little bit more minimal than they probably are in reality. And one of the things that comes out of that is that rather than there being, let's say, projections from left amygdala to right amygdala and other regions nearby the right amygdala, which is the reality, with a kind of exponentially decaying probability as a function of distance, we only have the connections between left amygdala and right amygdala. So coming back to Anil's simulations here, if we had a kind of richer expression of the cross-hemispheric connectivity in the connectivity matrix, then we would almost certainly see some more spreading in the stimulation in the contralateral hemisphere. So yeah, I mean, that would be an example of, that's all very contextual stuff. Like, I didn't tell you guys that. That's not the kind of thing that you would know without working in this field for a little bit. But there's a little bit, yeah, there's kind of context that comes into interpreting what we're seeing, and also interpreting what we put into the models in the first place. And that context it extends back in time, and it relates to the experimental techniques that are giving us these numbers that we're using to build our simulations from. And there are other kind of contexts that are relevant as well. So like if we're using diffusion weighted MRI tractography to build our connectivity matrices, which is a more common and just more or less better way of doing of constructing that connectivity matrix, there are experimental limitations and caveats that come with building your connectivities from that type of data as well, which I can go into. Tractography basically gives you lots of false positives, which is somewhat the opposite of the chemical track tracing scenario, which is a bit on the more on the false negative side. Excellent. So that's my run through of your guys' submissions. Very good. Very happy that you kind of picked up and rolled with things. Excellent. I have a comment here. You know, more comments, guys, more comments. We have some time to talk. Nice review. Is there a feedback, let's say, in the implementation of the models? Yeah, so there's a question here from Marcello Ramses around, it's really around the scientific method, I guess. Do we iterate in terms of what we're seeing in the models, and then go and do new experiments, and then kind of learn some more, and then refine and update our models? And yes, of course, we certainly do that. We don't do that enough, though. And that's something, I mean, personally, that's something that I'm trying to do more of. It's in computational neuroscience, it's, you know, you do have this situation that's the same in a lot of computational fields, where you have the kind of theoreticians and the real theoreticians, the like mathematicians. They're off in their own world. they you know, they're doing really important work in the area of finding new expressions for understanding the mathematical structure of things, but they're not like super fussed about direct connections to experiment, then like a continuum of different types of modelers towards people who are often more numerically oriented in terms of the simulation types as opposed to analytically, but who are trying to make like bidirectional connections between you know, novel experimental work. And it's something that, I mean, at the OCNS, which was just last week, which is the International Computational Neuroscience Society meeting, that was in the town hall, which is something you have in conferences where like people get together and do a kind of meta discussion on the conference and on the field. That was one of the things that was highlighted, and it's always highlighted, is, you know, to-do list, high priority, improve relationships with experimentalists. So I guess that was what the question that was coming from you there, Marcello, was. And the modest way that I'm trying to work in that area with my own research is, as I've mentioned, is I've been working on brain stimulation, also working on modeling EEG. And EEG is quite nice. Well, it's nice for a few reasons, but one of the reasons why it's a nice area to work on is that it's not the most expensive thing to do experimentally. So just at a practical level, it's easier to kind of run some experiments, do some models, work, see what you see, iterate, improve your models, new experiments, iterate, improve your models, new experiments. One of the issues, I actually say this a lot, but I don't actually hear other people saying it that much. So I don't know if it's an original point or not, or if it's just obvious, but neuroimaging studies are very expensive to run. They're more expensive now than they used to be, because we now know that we need larger sample sizes than we used to. So n equals 20 is no longer sufficient for neuroimaging. But bottom line is, you know, you need like $10, $20, $100,000 to do a neuroimaging study. So it kind of slows a bit that cycle, that theory experiment cycle. It makes iteration difficult, right? You basically need to design the study, run the study, get the results, and then that's 2 years of your professional life. So from a kind of, again, like a, say, a scientific method point of view, or a kind of even sociology of science point of view, I think that's really suboptimal. I mean, I think you really need faster iteration on theory experiment modeling than 2 years. It's really quite slow. So just my 2 pence on that there. Didn't know I was going to be sharing that with you guys. Cool. So again, let's have the questions coming through. Now I'm going to switch to the final wrap up part of this, which is just a comment briefly on what's in the other notebook that we haven't studied so much, which is modeling resting state. So as I said on Tuesday that one of the reasons why we're not covering it, in addition to not having enough time, is that it's a lot longer to run, this one. Actually, it renders better in MB Viewer, so I'm going to do it that way. A lot longer to run, and not longer to run because we're simulating multiple minutes of data rather than, as you've seen now in the simulations that we ran in the practical, 500 to 2000 milliseconds, right? So that's one of the things that is a bit different when you're trying to model fMRI data. So I'll just go through this. I'll go through this in the same way that I went through the last one, but I will be quick-ish. But so the idea here is to describe resting state, what we call resting state functional connectivity in fMRI. And so that is taking the time series of the bold signal, which fluctuate sample typically what we call the TR, the repetition time, in 2 seconds. So you have a data point every 2 seconds, and you'll record for 5 to 10 minutes. And you'll have these oscillating, kind of noisy oscillating time series for every voxel that you basically, three-dimensional pixel that you've recorded from. And then typically you do some kind of local spatial averaging, get some time representative time series for parcels from parcellation of the kind that we discussed, and then look at the correlation patterns between those regionally or locally averaged Pearson correlations. That's what resting state functional connectivity basically is. And the modeling approach is exactly the same as what we've talked about already. This is the connectivity, structural connectivity matrix here. Showing about these white dots in the plot. Actually, I should remove those. But this is the analog of, well, the one that I clicked off already, the connectivity matrix that we had before. But this is actually coming from tractography now, so it's a little bit more. So that's the length. This is the weight. You can still see a log scale, and there's these white things in the way, but that's our connectivity matrix. And then this is our fMRI matrix, so this is a functional connectivity matrix. What's different here is we have a cross here. The cross is because the ordering, I mean, what I showed you previously was a primary diagonal and then some secondary diagonals, a little kind of left to right diagonal in each of the off diagonal blocks. And in this case, you can see they're not going down kind of left to right, they're going up the middle. And this is just because the order's been flipped in the top half relative to the second half. I think because the authors who came up with this thought that this was a more symmetric and satisfying thing to look at. But it's the same thing. So these are the correlations between these brain regions. You can see the brain regions or the brain region abbreviations listed there. So this is the thing that we're trying to model. So just kind of picking a few things here, the approach that these authors took, and this is really a seminal work actually in the field now, the principal author here is Gustavo Deco, who is one of the top dudes in whole brain modeling, has made a lot of theoretical contributions and modeling contributions. And, arguably the field is now largely based on the approaches that he's pioneered over the last 10, 15 years. And one of those contributions is this mathematical model for neural activity that is targeted towards fMRI data. And it's targeted towards fMRI data because it's explicitly aimed at describing slow fluctuations. And the way in which this model is derived is it's pretty impressive, but it's basically doing some mathematical tricks on a what we call a spiking network model, a conductance-based spiking network model, and just taking the slow time constant components of activity in a what we call a leaky integrate and fire spiking network model, and some stuff that you don't see here because there isn't the derivation, it's just the equations. But what we end up with is this here, which is a one-dimensional differential equation, as opposed to the Janssen-Ripp model that we had, which was 6 dimensions. So there's a one-dimensional differential equation that's describing the evolution of this guy, the S, which is what we call a synaptic weighting variable, synaptic gating variable. And it's basically the slow part of neural activity. You think about it that way. This H here, just as an aside, is basically this playing the same role as the sigmoid function that we saw in the Janssen-Ripp model. And this final expression is actually just bringing in the network connectivity. So this C here is our connectivity matrix. So kind of summing the inputs from all of the other regions, which are the S's, and then also injecting some stimulation current, but we're not actually looking at the stimulation here. So that's the model. What the model looks like is, actually I want a time series plot. I think there's a time series plot. There isn't a time series plot in this. Should have put a time series plot in. We see, basically what you see is two kind of regimes. There's a low firing rate regime and a high firing rate regime. And the model basically flits between those two. It's bistable. You could kind of think of it as like a low energy state and a high energy state. And the way you know that it's bistable is for certain values of the parameters is in this phase diagram here. So this phase diagram is showing the value of the synaptic gating variable synaptic activity as a function of its derivative. So this is kind of how you do a phase plot for a one-dimensional system. And each of these lines is the value of the derivative for the corresponding value of the current value of the synaptic activity, and for a different parameter, the parameters being varied is the background input current. But the key thing, and we kind of have to zoom in to see it, is that for some values of this parameter, particularly the green line here, so this green line is basically intersecting 0 at 2 points. You can see it kind of comes in from the top, hits this dotted line, goes under, comes back, and then just intersects it, kind of only intersects it once. Yeah, it's kind of a continuous stand. So these two 0 crossings are what give the model its bistability. So we basically kind of flip between this state and this state, which is it's pretty interesting to see, and it corresponds to things you see in physiology. The next thing that we actually see here is a network level bifurcation diagram. So this plot here was about called the phase flow or the phase plane at the single node level. So remember, we have in our connectome-based network models, we have the nodes that define the dynamics, and then the network, the connectome that defines the global structure. So this is a node level thing, node level dynamics description. The bifurcation diagram here is actually a network level phenomenon. So it's actually looking at what is the basically, you can think about it as what is the stable state for different values of this parameter here, which is a global weighting on the coupling, on the multi-area coupling. And basically, as you're cranking up this bifurcation, what we call a bifurcation parameter, because we're seeing some bifurcations, then we go from there being a single stable state, and basically what that means is we have 80 nodes in the network, and at any given time, they're showing some, in this case, firing rate. So they're showing some kind of, think of it as an energy level, like I said, or the just a state, right? So roughly for low values of the global coupling, there's only one stable state in the network. But then eventually, when we cross a certain point, we have this bifurcation where we now see the continuation on the lower limb of this, of the low energy state that persists. But then we see the emergence of a high energy state or a high firing rate state. And then for every value of the global coupling going forward up until the second bifurcation here, then we have a bistable regime where we can either have a low firing rate or a high firing rate. And then again, we keep pushing up the global coupling parameter, which you can think of as like the energy, the system energy or something, or a kind of system heat type variable, because it's multiplying the weights between the nodes in the network, and there's no saturating nonlinearity in the couplings. So we keep pushing that up, and then eventually our low energy state loses stability and only our high energy state persists. So we have these two bifurcations. Okay. Now, the next thing that's interesting is that what they also then do is, and what we do in this notebook, is we study the fit of the empirical fit of the simulation to empirical data. So the empirical data, as a reminder, is this guy. So seeing how well we reproduce this guy, this matrix of Pearson correlations, as a function of different values of the global coupling. So the x-axis here is exactly the same as the x-axis here for different values of the global coupling. And what we can see is it's basically going up and up and up. So we're getting better fits to the data as we increase our global coupling up until a certain point, and then we drop. We have a little cliff over here. And the point where that cliff is, roughly within a little bit of error, is the location of the second bifurcation. Okay, so basically what this is suggesting, and this is something that is I guess, a loose, let's say, or a kind of generic theoretical idea that is, there are multiple versions of this in neuroscience and in this corner of neuroscience, but this is one version of it, is the idea that what we've got here is a bifurcation, and the zone around a bifurcation is typically understood as a critical regime, right? So when you get close to a bifurcation, system starts displaying interesting behavior. Depending on the type of system, the type of bifurcation, you have, things like critical slowing and magnification of oscillation amplitudes and so on. In this case, what we've got around the bifurcation area is the system is generating what you can think of as structured covariance patterns, right? So the data that we're matching earlier on, that connectivity matrix, that functional connectivity matrix, is a covariance structure. It's Pearson correlations between time series, but it has it's a structured covariance structure. Like it has this geometric, actually kind of physical structure that you can see in the matrix, and you have that kind of cross, and the arrangement of the nodes in that representation are they correspond to like locations in space. So it's a kind of spatially structured covariance structure. I like to say covariance structure rather than functional connectivity, but they're kind of synonymous. And that emerges in the model, or that is kind of maximized in the model around the bifurcation point, or the location of the second bifurcation, which is consistent with this idea that interesting stuff happens around bifurcations, broadly speaking, in neural systems. So yeah, we can take a look at how the simulation is doing in terms of the, like at that optimal point. This is the model. So one of the things that actually these type of models, they're getting better. This was from 2012, but still recovering these cross-hemispheric connections, that's what the line going up here, right, is. Recovering this part of the correlation structure is something that these models don't always do a great job of, and they're definitely not doing a great job of that here. So things like the correlation between left and right amygdala, for example, is definitely underestimated in the simulation here. But broadly, like the correlation is not bad as far as the type of what we kind of tend to look for here, like 0.4 to 0.6. The best that you can generally hope for is around 0.8 for these type of things, and 0.4 or 0.5 is not a bad job. Cool. So yeah, the other things, these are just actually reproducing some figures from the paper, like a kind of vector, column-wise plot of one of the columns from the connectivity matrix, which we don't need to go into the details here. But yeah, so as I said, this is quite an involved tutorial, especially if you kind of get into the nuts and bolts of the bifurcation diagrams and so on. I give you a rapid run through, as opposed to doing that for an entire session, and hopefully that kind of filled out a little bit some of your understanding of the type of things that we can do in this area. And you have the references here, right? So if you're interested in this, actually there was a question in the chat from day one, which was, can we have some references? So there are references in this end of this notebook. The key one is this one. So this is the study that this notebook is actually reproducing, and this is really a classic paper. There's a lot in this paper. It's very, and some of it is quite advanced, but it's also, it's not kind of impenetrable at the same time. This final one as well is actually a follow-up to this one, which expands the model and looks at things like time varying correlation patterns, which we haven't looked at here, but is also a big theme in the field that generally goes by the name functional connectivity dynamics. And this idea of having switching behavior. So you kind of switch between certain network configurations over the course of minutes in these correlation patterns. So that's something that people are very interested in modeling these days. Great. Okay, so I'm going to call it a day now, guys. We have one more question in the chat. So the question is, with respect to bifurcations, very stable, bistable, i.e. it could be some dispersion in the two attractors, but does not show during the coexistence of the high-low energy states. Is this due to the nature of the model? So dispersion in the attractors does not show during the coexistence. Let me just think about that for a second. So I think by dispersion in the two attractors, you mean kind of separation two attractors?
Right. No. Well, maybe I can explain, John, if
Go
you
ahead.
don't mind.
Yeah,
So
thanks.
what I mean is, like, you see these low and high energy states, right? And usually you should do bifurcation models of a logistic model, for instance, right? You see that the attractors converge to the given value. But in this particular case, I don't know if it is due to the sample that you are showing or what, it's very sharp, right? You don't see any dispersion, any oscillatory behavior towards the attractors. It's just cut into high and low energy. They basically bifurcate almost immediately. So I was wondering if this is because the way the model is created. And I think you mentioned something related to that because of the way the connectivity matrix works. But I was wondering if you have any comments about that.
So you're talking about a bifurcation diagram that looks like a fork?
Yes,
where you have the low and they get further away as the energy goes up.
Right, and at some point you may see some oscillations around these forks, these two branches, right?
Yeah. Well, I think the main reason why this is different is that we have like the low energy, so there's kind of a baseline, there's a floor, so it's not, there isn't really any room for you to go on the lower limb because you're near zero.
Right.
So I mean maybe if you scaled it differently, you might see that actually. Now I think about it, but I'm not 100% sure. So yeah, I'm not kind of, no examples really spring to mind of, equivalent logistic map type models that have that, have that kind of flaw but maybe they're a physical system, like well-studied systems that do show that as well. I understand what you're saying, but I mean, we've still got that phenomenology, right? We're moving away, and as we move away, we're kind of diverging, right? It's just we're not diverging on the lower part. Or this is staying constant. I guess you were maybe saying two things, weren't you? You were saying this isn't moving continuously. This upper limb isn't necessarily moving continuously from here, and also this lower limb is not also diverging. It's staying constant.
That's right. That's right. I just posted a link in the chat. So if you open it, you will, I guess you will see what I'm talking about. So that's kind of the logistic map bifurcation diagram that you usually see. But then you have this oscillatory behavior, right? And sometimes this is due to different dynamics, like chaotic dynamics, when the solution jumps between branches, different stable branches. So you don't see anything like that in your model. And you're right. In one case, the low energy state is just constant, and it makes sense because it's the low energy state is just zero, but the other basically increase a little bit, but you don't see any kind of random behavior like the one that you see at the bottom of this diagram, for instance, right?
The bottom or the right hand side?
The last part, sorry, the right hand side.
Oh, right. I mean, that's a good question. I think, to be honest, like we're not going that far. So we could explore what happens on the right hand side. My intuition, I have to say though, is that we wouldn't, it wouldn't go crazy because there aren't any, in this model, there aren't any squashing functions. And there aren't many nonlinearities either. So I think it's just going to explode rather than move into a qualitatively different regime, if that makes sense.
That makes sense. That completely makes sense. The nonlinearity, for sure, that makes sense, for sure.
And there's no explicit negative feedback in the in the dynamics. It's really just it's
propagation and diffusion mostly, right?
Like a Brownian motion basically around the attractor. It's just fluctuation,
diffusion.
So exactly. So it's not, and so there's no like genuine oscillations there either, because
Right.
There's bistability, but it's not an oscillation in the sense of an oscillation generated by negative feedback.
And this is mostly justified because it's the steady state solution that you're looking for in this case, or the rest state.
Not necessarily. I don't think the model was derived like explicitly with those kind of assumptions. I did mention those in the first lecture, and there are other, like linear graph diffusion type models that are explicitly describing steady state behavior, but the model in this case is not, it's not linearized around a fixed point.
Okay.
It's just a reduction. It's a reduction of a more complicated and more nonlinear model, but the main focus of the simplifications is getting rid of all the fast stuff, getting rid of all the fast fluctuating parts of it, and then the slower ones, the slowly fluctuating modes or the slowly fluctuating time constants dominate the behavior on the longer time scales that you're looking at with fMRI. Like they dominate the dynamics on the order of seconds. So everything that's going on with, you know, a kind of after a spike, like the back and forth of excitatory and inhibitory activity that's going on the order of milliseconds, that's all just what in the wash. When you're looking at things on the order of seconds, you're looking at slowly varying things, and that's the rationale behind the derivation of this model. But it's used to study resting state behavior, nevertheless. Yeah, it's used to look at steady state activity.
Nice, very interesting. Thanks.
Yeah, thanks for the questions. Good questions. Okay, guys. Well, I'm gonna wrap up. It's been a pleasure going through this, and I hope you guys found it valuable. Thanks a lot for your efforts in the homework, and you know you were thinking hard about some of the interpretations and coming up with some interesting things to do with the things that I handed you to without an awful lot of hand holding. So good job there. And if you want to get in touch with me, if you feel like I haven't explained stuff very well, if you want to see more, there's plenty more to see, then reach out. I think you may have my email already, but if you don't, well, I'll put it in the chat right now. This is the best one to reach me on. So you can contact me there, or you can grab me on Twitter at that handle, and you can check out my website for stuff that we're doing, get a bit more context on where I'm coming from here. So thanks a lot for your attention. Oh, I sent those privately. Okay, I'll resend those. That's my email. That's the twit. That's the website. Okay, had a bit of feedback. Thanks. Okay, thanks guys for your attention. You know, I feel like it's a bit strange speaking into the void, so I hope it hasn't been too unusual, or I haven't seemed too distracted, because I haven't had, you know, a bit of eye contact to get the vibe. I just have to imagine it. Right. So you know where to go if you want to follow up, guys. Have a great day. Goodbye.